A very happy afternoon, students. I welcome you back once again on behalf of RGR Academy, and I welcome to the biology session. I hope you're all safe and sound at home. Let us begin with this session. Fine. The last class we discussed about the photosynthesis. As we begin with the plant physiology. The plant physiology, the first chapter, we begin with the photosynthesis. And the photosynthesis, we discussed about the different types of the cycles that we started already, and we almost like completed that particular chapter. As far as the photosynthesis is concerned, that is one of the important chapter. As far as the plant physiology is concerned, we expect like 4 to 5 questions from that particular chapter, that is photosynthesis itself, where we discussed about the different pathways. Yesterday we discussed C2 pathway, that is the photorespiration, and C3 pathway, that is Calvin cycle, and C4 pathway, that is Atch and Slack pathway. So, and the special type of anatomy that we discussed, the anatomy is Grant's anatomy, along with the mesophyll cell, the contribution of the bundle sheath cell, that also we discussed yesterday. So, as far as this chapter, I mean, this particular unit is concerned, the plant physiology is concerned, as already we discussed, give more importance for a few chapters out of like 6-7 chapters that you could see in this particular unit. So my suggestion is to give more importance for chapters like photosynthesis, number 1, and the 2 is respiration in plants. You can give again importance for the respiration in plants. And coming to the transport mechanism, you can also give importance for the nitrogen cycle. The nitrogen cycle you can study, the nitrogen fixation, and root nodal formation, the nitrogenous enzymes that functions that you can study, and pathways like the transpiration and apoplast, symplast, these type of pathways that you need to study, and active transport, passive transport, facilitated transport, right? How the difference it makes between these types of transport. There's also like areas that you can get questions from this particular chapter. And coming to the next part, the respiration in plants. In this part, you mainly be studying about aerobic and anaerobic respiration. So coming to aerobic respiration, glycolysis, right? You study glycolysis, tricarboxylic acid cycle, that is Krebs cycle, electron transport chain, then oxidative phosphorylation, then pyruvate decarboxylation that you'll study, and also, right, we'll wind up with the anaerobic type of respiration, fermentation, alcoholic fermentation, acetic acid fermentation, lactic acid fermentation. These are different types of fermentation reactions which occurs, the pathways as far as anaerobic respiration is concerned. So, and also amphibolic pathway, then pentose, that is like pentose pathway, exose monophosphate pathway that we call it as, and the respiratory quotient, and the balance sheet of ATP utilization, right? And these are the main areas that we cover in this particular chapter. And from this particular chapter, respiration plans, you can sense, even though it looked like very small chapter, but the questions, like, often you get questions from this particular chapter, you get maximum of 3 questions you get from this particular chapter. So today we'll discuss about, I will try to complete this chapter, respiration in plants. Apart from this chapter, we can also study the phytohormones, the plant growth factors, regulators like auxins, gibberellins, cytokinins, abscisic acid. These are the various hormones that we'll study. And so collectively, this plant physiology also like very important unit as far as the Neet preparation is concerned. So as of now, we completed 1 chapter, that is photosynthesis we have done. So I hope you guys, you revised what all things that we discussed yesterday, especially C2 pathway, C3 pathway, and C4 pathway, the difference between C3 plants and C4 plants, photosystem 1 and photosystem 2, cyclic and non-cyclic photophosphorylation, that is Z scheme, and pigments especially, that are for photo reactive pigments, photo complex quantosomes, these areas. And these are all the things that we discussed yesterday for your kind of information. So if you didn't attend the yesterday's session, like, this is a like recap for you what all the things that we've done so far as far as photosynthesis is concerned. So, and we also concluded that chapter with the CAM pathway, crustacean pathway, right? This particular pathway is also like, where we discuss is vice versa conditions like stomata. We discuss something like sunken stomata, where the stomata is actually closed during daytime and it is open during the nighttime. This usually you could see in case of like xerophytes or desert plants, this type of mechanisms where try to conserve the water. And the advantages of Hatch and slack pathway that we discussed. So on the whole, the collectively the photo respiration, the C3 and C4 cycle, which all like which is gain like advantage, so that you can gain marks from this chapter. So you can get questions from any part. But today we'll start with the next chapter, that is respiration in plants. So let's begin with this chapter without any delay. So we begin with the respiration in plants. Coming to the respiration in plants, we'll start with the respiration. So respiration in plants. In the last chapter, the photosynthesis, we discussed, because the photosynthesis, which plays a very important role in the energy, in the formation of like how the energy is getting produced. So that was that we discussed. So how the energy is going to get liberated in the plants by means of ATP, that is adenosine triphosphate, that is energy currency of the cell. So in this chapter, we're going to discuss, already we discussed how the energy is produced. Energy is produced by means of the photosynthesis. Then now we're going to discuss how this energy is going to get consumed, how the energy is going to get liberated, how the energy is going to get disseminated inside the plants and the cells that we're going to discuss, in which we'll discuss various pathways like aerobic pathway and anaerobic pathway, aerobic respiration or anaerobic respirations, all those things that we discuss in this particular chapter. Because energy, so how much ever the energy is produced by means of photosynthesis, this has to be produced in a controlled manner. It has to be produced in a systematic and controlled manner. How this energy is getting produced by means of adenosine triphosphate, and this type of respiration which occurs in a cell, we call it as a cellular respiration. So the cells which playing very important role in this type of respiration, so we call it a cellular respiration. So now the energy is getting produced. The energy is in the form of what? Energy is in the form of ATP, that is adenosine triphosphate. We call it as energy currency of the cell. So this respiration which occurs in the cellular mode, so we call it a cellular respiration. We call it a cellular respiration. Coming to the cellular respiration, so we make it a simple form like C6, this is a glucose, C6H12O6. This get combines with 6 molecules of oxygen, and this gives rise to 6 molecules of carbon dioxide, but 6 molecules of H2O, and the energy is going to get liberated. This is a simple way by which we can make it like the cellular respiration. When the glucose is getting converted to 6 carbon dioxide and 6 water molecules, and the energy is going to get produced. So what is going to happen here? The complex form is going to get converted into the simpler forms. Of course, the complex form is going to get converted into the simpler form by means of the cellular respiration, right? This cellular respiration we're going to discuss. This respiration can be aerobic or anaerobic. This respiration mechanism can be aerobic mechanism, or this can be anaerobic. So in this chapter, we discuss both about aerobic and anaerobic respiration. Coming to the aerobic respiration, you know the couple of examples about what we discussed in aerobic respiration. Coming to aerobic respiration, the aerobic respiration. Can you give some examples for aerobic respiration? Examples for aerobic respiration. The first example is that the couple of things that we're going to discuss here. Glycolysis. Then after the glycolysis, what are things we're going to discuss here is the breakdown of the pyruvic acid. Breakdown of pyruvic acid. Then Krebs cycle, that is TCA cycle, Krebs cycle. Then the fourth one we discuss about electron transport chain. These are the 4 important things that we're going to discuss under the aerobic respiration. So glycolysis, breakdown of the pyruvic acid, then we have the Krebs cycle, electron transport chain. Also we're going to discuss about like oxidative decarboxylation. We also discuss about oxidative decarboxylation. So oxidative decarboxylation we're going to discuss. So these things which comes like, along with this, right? Along with this, we have like oxidative phosphorylation that we'll discuss separately. Oxidative phosphorylation, that is end part of electron transport chain. We discuss about oxidative phosphorylation. And also HMP, exose monophosphate pathway. So these areas that we're going to cover: glycolysis, breakdown of pyruvic acid, Krebs cycle, electron transport chain, oxidative decarboxylation, oxidative phosphorylation. Oxidative, if it is my writing is not clear, I'll just repeat this: oxidative phosphorylation. Then we have HMP. HMP stands for exose monophosphate pathway. There is other name for HMP also, that also we will discuss. We call it the triple P, right? Proto for pentose pathway, right? So that also we going to discuss, like HMP. And these all comprises of that aerobic respiration. Can someone tell me, like, how many ATPs usually get, get liberated? The net gain of ATP during aerobic respiration. What is the net gain of ATP? Total, the net gain of ATP. I'm not asking for gross, I'm talking about net gain. How many ATPs actually you could see the net gain as far as aerobic respiration, and how many ATPs which seems to be a net gain in anaerobic respiration? Simultaneously, we have like other type of respiration, we call it as anaerobic respiration. In this type anaerobic respiration, this process which usually occurs in the absence of oxygen by means of fermentation. This occurs by means of fermentation. We have different types of the fermentation. You study the fermentations usually which occurs by means of the Saccharomyces cerevisiae in brewing industry, baking industry, in all these places like wine, all these confectionaries, that you could see the fermentation process which occurs in the absence of oxygen or in the presence of the carbon dioxide. So this type of respiration we call it as anaerobic respiration, where this respiration, right, we have alcoholic fermentation, we have the alcoholic fermentation, we have lactic acid fermentation, we have acetic acid fermentation. We have these types of the fermentation, I mean the reactions which occurs during in the anaerobic mode of respiration. So fermentation, we have alcoholic respiration. I mean the ethyl alcohol, the glucose molecules, like the sugars, the glucose molecules is getting broken down into the simple thing, and you could see the product like ethyl alcohol, where you could see alcoholic fermentation. In case like lactic acid, that is La stands for lactic acid fermentation. In this case, lactic acid fermentation, you could see the bacteria. Can you name the bacteria which is responsible for the production of lactic acid? What's the name of the bacteria? We call it as LAB. What is LAB? LAB stands for this responsible for the production. Usually this particular bacteria which acts on the casein or the milk protein which produce lactic acid. Can you name what is the name of this bacteria which is responsible for the lactic acid fermentation, I mean production of lactic acid? Coming to this acetic acid, acid also you could see the kind of bacteria. Can you name the name of a bacteria, the bacteria which is responsible for acetic acid? So LAB stands for lactic acid bacilli. Lactic acid bacilli, it's a correct answer. Yes, correct answer, lactic acid bacilli. And acetic acid, coming to acetic acid fermentation, can you name the microorganism, the bacteria which is responsible for acetobacter? Yeah, it is Acetobacter aceti. Coming to the acetic acid, Acetobacter aceti is the bacteria which is responsible for acetic acid. How about alcoholic fermentation? Alcoholic fermentation can happen by means of bacteria or commonly by means of yeast. Can you name the yeast which is responsible for the alcoholic fermentation? Can you name we commonly call it as a baker's yeast. Baker's yeast. Can you name which is called the baker's yeast, which is responsible for the alcoholic fermentation in a beer and wine and this type of industries where you could see, right, this type of like yeast which is responsible for this type of fermentation, right? That is the reason why this yeast is commonly called as baker's yeast. Can you name the name of the yeast which is responsible for this alcohol fermentation? Exactly, Saccharomyces cerevisiae. Saccharomyces cerevisiae is the name of the yeast which is responsible for ethyl alcohol production or the alcohol fermentation. And lactic acid is a lactic acid bacilli, and acetic acid is the Acetobacter aceti. These organisms which goes to special mode of cellular respiration in the absence of oxygen, so we call it as anaerobic respiration. So together we call it as a fermentation process. So this first outline, the introduction of this chapter within cellular respiration, we categorize the respiration into two types. One is aerobic respiration and other one is anaerobic respiration. In aerobic respiration, the net gain of ATP, I asked this question, many of you gave the correct answer. Coming to the aerobic respiration, the total gain of ATP is 36 ATP. So you need to be very careful. It's some people you mention as 38. Its total gross is 38, but net gain is actually 36. So out of 38, what happens to ATP which is get lost actually will be utilized back. So the net gain during aerobic respiration is 36 adenosine triphosphate. Coming to anaerobic respiration is concerned, the net gain of ATP is just only 2. Anaerobic respiration, you could see only 2 ATP molecules in the anaerobic cellular respiration. If question comes in this way, what is the gross? If they ask for the gross ATP, then it is 38. Gross is 38. But if you ask about the net gain in aerobic respiration, net gain is 36. But the gross they ask, if the question comes like gross, the gross is 38. In which what happens? 2 ATPs which is getting actually utilized, taken back. So 38 minus 2, it is 36. So the 36, this 36 is the net. This 36 is the net gain ATP in the aerobic respiration. So remember the question, if they ask us about the gross, or they asking about the net. If the net gain, it's a 36. If it is a gross, they're asking it is a 38. We're going to discuss like how this 36, we're going to discuss about the balance sheet as well. So how this 36 which is coming all together. At the end of this chapter, we're going to discuss detail of the balance sheet of the ATP production also. Coming to the anaerobic respiration, 2 ATP molecules, just only 2 ATP molecules that you could see in case of anaerobic respiration. So if this area, if you get a question, right, anaerobic respiration, sometimes anaerobic fermentation, you get like what is the name of the bacteria that is responsible for lactic acid? You can answer the lactic acid bacilli. Acetic acid is Acetobacter aceti. Acetic acid is by means of Acetobacter aceti. Acetobacter aceti is the name of the bacteria. And come to alcoholic fermentation or ethyl alcohol, it's the Saccharomyces cerevisiae or the baker's yeast. So this is just an outline or introductory part of the respiration in plants. Fine. So now we move on to the first part, that is an aerobic respiration. Let us begin with the glycolysis. We begin with glycolysis, the first aerobic respiration. So as you all studied in the textbook, at this point of time, you must be aware of like glycolysis process. Yes, we continue, but I'm not going to explain in detail each and every step. But questions they'll ask in the NEET examinations, like any just one part or one segment of the glycolysis they take, and they'll ask what is the name of the enzyme, right? So any particular the segment of the glycolysis, they particularly any one segment, they just take it and splice out, and they'll ask what is the name of the enzyme which involved in the process. So this type of questions that you can get from the glycolysis, or the net gain, they'll ask are they gross, the ATP production, that they will ask from this particular pathway, that is glycolysis. When coming to the glycolysis, there are two pathways like that you could see, the two segments. This segments like from here, from glucose, from this area, this is the preparatory stage. This is a preparatory stage, and where like the energy is getting manufactured, the energy is getting prepared, and the energy is getting prepared, and the energy is getting liberated. In this case, the energy is getting produced. So first half of the phase is the preparatory phase, and the second phase actually of the glycolysis, we call it as a payoff phase. Yeah, you're correct, payoff phase. And second phase is actually constructive phase, where you could see 2 ATP molecules. So here you could see, here is one ATP, and here also you could see one ATP. So how this occurs, where the glucose molecules is actually getting the glucose molecule that you could see. What is the product here? The product here is pyruvic acid. The product is pyruvic acid. Yes, guys, you're correct. Payoff phase is the correct answer. So pyruvate phase. I mean pyruvic acid that you could see at the last. But how many pyruvic acid? One glycolysis which gives like one pyruvic acid or two pyruvic acid. So how many pyruvic acid which is getting produced out of glycolysis? One or two. But this diagram usually in the textbook or whichever the images that you could see, right? You could see 2 pyruvic acid. So the same process, whatever the the schematic thing that you mentioned, the flow chart, it's also applicable on both the sides. On both the sides, you could see the same thing, and here also you could see 1 pyruvic acid. And one more thing, the glycolysis, which can be also called as EMP pathway. The glycolysis, which can be also called as EMP pathway. Can someone expand what is EMP? EMP is actually the name of the contribution of the scientist. So what is the EMP expansion, EMDN, it starts with EMDN, right? So what's the expansion of EMP? Glycolysis can also be called as EMP. Fine. So when you coming to this process of glycolysis, you could see 2 pyruvic acid, but you should understand always, like, most of the times when the calculative part comes, you usually forget the other pyruvic acid on the other side. So one glucose molecules which produce 2 pyruvic acid. So for 2 pyruvic acid on the both the sides, one is from the glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate, and other is one dihydroxyacetone phosphate, DHAP, dihydroxyacetone phosphate, and from the glyceraldehyde 3-phosphate. So from both the sides, you could see the production of the pyruvic acid. Emden-Meyer pathway. So the expansion of EMB is Emden-Meyer pathway. Okay, Emden-Meyer pathway. Up Emden-Meyer Parnas. Yeah, P stands for Parnas. Emden-Meyer Parnas pathway. For this, guys, they contributed in such a way. So on behalf of their contribution, this glycolysis process can also be termed as EMP pathway. So understand, 2 pyruvic acid is getting produced here, and glycolysis is an oxidative process. So sometimes they'll ask, like, what type of process is this? Glycolysis is an oxidative process where one glucose molecule is getting converted to product. What is the product here? Pyruvic acid. 2 pyruvic acid is getting formed. And where the glycolysis process occurs, which part of the cell? Can someone tell me? This also sometimes they'll ask you questions. Which part of the cell where the glycolysis process takes place? Which part of the cell where they're given options, and you need to identify which part of the cell the glycolysis takes place. In cytoplasm, exactly. So in cytoplasm or cytosol, where you could see this glycolysis process takes place. And glycolysis is an oxidative process where one glucose molecule gets converted to 2 pyruvic acid. So another question they'll ask, it's an aerobic process or anaerobic process? Can someone tell me? Glycolysis is purely aerobic, or could you see a glycolysis in anaerobic state also? Can you justify glycolysis? Is this the aerobic process or it is an anaerobic process? So what type of process is this? Glycolysis is aerobic or glycolysis is anaerobic? So this is also like sometimes, like, glycolysis is purely aerobic or glycolysis is anaerobic. So both. Glycolysis is both aerobic and anaerobic. So when the situation comes, like, you be in dilemma, like whether which one I need to select. So glycolysis is both aerobic as well as anaerobic, and this is an oxidative process, right? So what happens? What is actually the gross gain if altogether, if you see, like, aerobic process, I mean aerobic respiration, the net gain is what? Net gain is a 36 ATP. Just if you see only in a glycolysis, only in glycolysis, what is the net gain? What's the net gain of ATP only in glycolysis? You got the question? What is the net gain of ATP? Net gain of ATP only in glycolysis. On the whole, aerobic respiration is 36 ATP net gain. So only in glycolysis, what is the net gain? If this type of question comes also, you must be in position to answer. Understand, like, both the sides, like here also you could see 1 ATP. See here you could see 1 ATP. Here you could see 1 ATP on the other side. So this side also you could see 1 ATP, and this side also you could see 1 ATP. So on the whole, what you could see is, what are the products that you get at the end of the pyruvic acid? Let me make it out. So as far as the calculated part is concerned, I'll make it in this way. So glycolysis. So first we're done with the glycolysis. So what are things that we received? We got 2 pyruvic acid out of the glycolysis. And how many ATPs which has been formed? 4 ATPs has been formed. So what are things that we received out of the glycolysis? 2 pyruvic acid, yes, and 4 ATP molecules has been formed. And out of this 4 ATP molecules, and 2 molecules which has been ATP molecules which has got consumed already, which has got consumed. So 4 ATPs has been formed, and out of 4 ATP, what has been happened? 2 ATPs which has already got consumed. So remaining is only 2 ATP. So the net gain in the glycolysis is just only the 2 ATP. It is not 4. When you gain, when I asking about the net gain is what? Net gain is only 2 ATP, because two ATPs which has already been consumed in the beginning, the first phase. So you could see only the two ATP, that is a net gain. And what happens, there is a remaining six ATPs also there. But this six ATPs, which actually is in the form of what? Which actually in the form of 2NADH2. 2NADH2. This gives 6 ATP. So 2NADH2 has been produced in the glycolysis process. You know, one NADPH is equal to how many ATP? One NADPH2 is equal to 3 ATP. So 2 NADPH2 is produced in a glycolysis. So what is happening, like, so one NADPH, you need to understand, one NADH2 is equal to 3 ATP, right? So 3 ATP. So 3 into 2, this gives the 6 ATP. So we have the 2 ATPs here and we have the 6 ATPs. So what is happening now, where you can sometimes you have these questions like, whether I should combine this 2 plus 6, or I can make it as 8, or I should mention only as a 2 ATP. So which one is the correct answer? So what is actually happening here is, out of 4 ATP, 2 ATPs which has got consumed. So the net gain is only 2 ATP. But remaining 6 ATPs have been formed in the form of 2 NADH2. So 1 NADH2 is equivalent to 3 ATPs, right? So 2 NADH2, so 3 into 2, that is equal to 6 ATPs. But what's the fate of the 6 ATP? The 6 ATPs, where this will be like a check. It's not like a cash. This 6 ATP is like a check form. So what the check, like, you need to deposit the check in a bank, and later part only this will be encashed, right? See, this is the form of a check. But so now the cell cannot utilize the 6 ATP at this moment. The later part, that you see, like, this encashment occurred during oxidative phosphorylation. During oxidative phosphorylation, the 6 ATP which can be withdrawn, which can be encashed. So now we cannot do anything with the 6 ATP, because the 6 ATP is like a cash form, that is which has been produced by NADH2. So as if now the net gain is only what? The net gain is only 2 ATP. But on the whole, how many ATPs are produced in the end of glycolysis is 8 ATP. In that way, it is 8 ATP. But gain is only 2 ATP. Why 2 ATP? Then what happened to remaining the 6? The 6 actually is in the form of a check. It will not be actually encashed now. This will be encashed during the later stage, that is an oxidative phosphorylation. This 6 ATPs which will be encashed. So this concept that you should understand clearly. Okay. Coming to the glycolysis, I'll just move on to the previous part of the glycolysis so that you can see. See, in this case, here you could see, here ATP, 1, 2, 3, 4. You got 4 ATPs here. So you got, how we got the 4 ATP? 1, 2, 3, 4. So we got like 4 ATP. Out of this 4 ATP, like 2 ATPs by means of like shuttle system, like, this 2 ATPs which are going to get consumed. So net gain is only 2 ATPs. So 4 here, and you could see NADH2. Here is 1 NADH2. Here also you could see 1 NADH2. So 2 NADH2. So I told you, like, 1 NADH2 is equal to 3 ATP. Any 3 ATP, right? So 3 ATP is actually, which is getting formed out of one NADH2. You understand? 1 NADH2 is equal to 3 ATP. The same way FADH. When you talk about FADH2, like, this is equivalent to 2 ATP. This you should understand. Whenever like FADH2 comes, like, 1 FADH2 is equivalent to 2 ATP. 1 NADH2 is equal to 3 ATP. This will be beneficial when you sum up everything, like in the balance sheet when you make. So 1 NADH2 is equivalent to 3 ATP. 1 FADH2 is equal to 2 ATP. Okay, 2 ATP. So here there is no FADH2 here, but you could see only the NADH2. So 2 NADH2. So 1 NADH2 is equal to 3. So 3 into 2 is equal to 6. And you got the 4 ATP. I told you, like, out of 4 ATP, that is 4 minus 2, which is happened, like 2 has been utilized. So the net gain is 2. So total is actually, you can say it is 8. But what happened to the 6? The 6 is like in the form of a check. It will be encashed only during oxidative phosphorylation. So this 6 ATP is not going to utilize in the glycolysis process. Yes, it's produced in the form of NADH2, but this is the form of cash. Sorry, this is the form of a check. When it will be encashed, this will be encashed only in oxidative phosphorylation. That is the time where the ATP, the purpose of ATP, which will be completely fulfilled. So guys, the total ATP when it comes, it is 8 ATP, but the net gain again, if the question comes, so it is only 2 ATP. This question is like sometimes there'll be tricky question. Please be very careful. If they ask about the gross, or are they asking about the net gain, or they asking about the total ATP, so you need to be very careful in when you just read the question as far as glycolysis is concerned, because you're getting different options. Like you get an 8, you get sometimes get 2, you get like 4. Just read the question properly and then you answer the particular question as far as glycolysis is concerned. So understand, guys, the net gain at the end of the glycolysis is only the 2 ATP, but the total ATPs which is actually produced is what? Produce the 8, but out of 8, 6 ATPs, actually it is 6 ATPs a form of a check. This will not be encashed. This will be utilized only during oxidative phosphorylation. Gross ATP is 4. Gross ATP is 4. Out of this 4, 2 ATP is going to get utilized. So it's going to get utilized, then 2 will be the net gain. So when they ask about the gross ATP is a 4. Out of 4, 2 will be utilized and 2 will be the net gain. So remember the question. So if read the question properly, and sometimes you also like should be very careful and extra cautious when the glycolysis process, where the one side the pyruvic acid is like it's getting produced, you need to be careful. Like other side also the same reaction is produced, is getting formed. So one glucose molecule which produce 2 pyruvic acid. It means 2 pyruvic acid means 2 pyruvic acid which goes to the 2 Krebs cycle. That is also you should be very careful, because once you're done with the glycolysis, don't think like the pyruvic acid just it's produced. But what is the fate of this pyruvic acid? The pyruvic acid then again it get converted to acetyl CoA, right? Decarboxylation takes place. Pyruvic acid which get converted to acetyl CoA. That is one stage, and after that what will happen? Acetyl CoA which goes to the Krebs cycle. The Krebs cycle, the tricarboxylic acid cycle. So 2 places here also one Krebs cycle takes place from here, and this side also another Krebs cycle takes place. This area, right? You sometimes like you need to be very very cautious. What type of question? Whether they're asking the question from the pyruvic acid, are they asking question from the acetyl CoA? When we discuss about the Krebs cycle, you should be very very careful and very cautious, because the question sometimes I'll ask accordingly. You need to calculate whether they're asking a question right from the pyruvic acid or they're asking only from the acetyl CoA. So that we're going to discuss in the next part, that we'll discuss in a Krebs cycle. But in the first stage, the glycolysis, you should be very careful, guys. So what is the net ATP, what is the gross ATP, and how many ATPs got consumed, and how many ATPs actually in the form of check. I mean, it's not utilized. There are only 6 ATPs, because the 6 ATPs is just formed out of NADH2, because 1 NADH2 is equivalent to 3 ATP, 1 FADH2 is equivalent to 2 ATP. So we're going to just multiply that into 2. So you're getting 6. This 6, when the 6 ATP is going to get utilized, even though the ATPs are produced in the form of NADH2, yes, but there's no use as if now, because it's going to get utilized, its function going to get fulfilled only during oxidative phosphorylation. I hope you're clear with this process. So we'll photolysis. So you ask like what happens during the water loss. Water loss is again is by means of photolysis, where you could see the chemiosmotic hypothesis. We have shuttle system that we'll discuss in the next part. The shuttle system we have 2 types of shuttle system, malate and we have aspartate, the shuttle system. That shuttle system which takes care of the water loss. That is a continuation of the chemiosmotic theory, because we discussed already about the chemiosmotic theory. This part also we discussed when we come to the 5th complex of electron transport chain, that is FO, F1. That part when we come, we'll discuss about the water loss and how this protons is getting like transferred, that we'll discuss. Okay, so I hope you're clear with the glycolysis process. Guys, from this area, how do you get a question is, either you will not be getting a full part, either they get what is the name of the enzyme. So what are the monomers? If the question comes like, the monomer part they ask. So what are the monomers that you could see as far as the glycolysis is concerned? So G3P, these are the monomer. They ask about the monomer sometimes. If the question comes like, what are the monomer? Glyceraldehyde 3-phosphate is a monomer. DHAP, dihydroxyacetone phosphate. This is the monomer. So they give the options. They'll ask you to identify the monomers in the glycolysis. G3P, glyceraldehyde 3-phosphate is a monomer. Dihydroxyacetone phosphate or DHAP this is also the monomer again. And fructose 6-phosphate, and glucose 6-phosphate, this can be also acting like a monomers. So coming to the glycolysis, you can get a question from any part, not only from the ATP gain or loss, but also get a question from these areas like the monomers. Glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, fructose 6-phosphate, glucose 6-phosphate. These are all monomers. If question comes like find out the monomers from the glycolysis. If you could see any of these options, then you just go fructose 6-phosphate, glucose 6-phosphate, G3P, and DHAP. These are all the monomers. And they'll ask actually, like, functions of enzyme kinase. You could see the enzyme kinase. They'll ask the question, and the function of phosphorylase. So the main feature of kinase and phosphorylase, this can either consume or it can produce ATP. Dual function. The kinase and the, you could see the kinase, right? So you could see the kinase here. The kinase, and you could see the isomerase. These enzyme which plays a dual role, which can consume, the kinase enzyme which can consume or it can produce ATP. This both the functions, dual functions, this plays by the this particular enzyme, the kinase. Okay, this area also you can get a question. So kinase or phosphorylase, these two enzymes act like it can consume, at the same time it can also produce the ATP. So glycolysis is concerned, you can get a question from any point of time. Don't think just only if you study the flow chart it is quite sufficient, but you need to understand the ATP productions, how the ATP is getting formed, and what is the functions of these enzymes, or one particular segment, any one particular segment they'll ask, and they'll ask you to identify what is the name of the enzyme, how many ATPs is being produced, how many ATPs got consumed. So these are all the two ATPs where it is going to get consumed. These two ATPs are going to get consumed in the shuttle system that we'll discuss. That area also during glycolysis, two ATPs are consumed, that we discussed -2, right? So where it is getting consumed, how the ATP is getting consumed, that way also they ask a question. This is getting consumed by means of shuttle system, that we'll discuss. Fine. So I hope you're clear with this part, the first part of the aerobic respiration that we discussed about the glycolysis. Second, we discuss about breakdown of pyruvic acid. The second part of the aerobic respiration will start with the breakdown of pyruvic acid. Now, what the end product of the glycolysis? The pyruvic acid. Now, the pyruvic acid which goes to the next part, this is called as a breakdown of this pyruvic acid supposed to break down, right? So what is this process is called? This process is called as the decarboxylation process, which is going to occur. So coming to the decarboxylation process, let me show this diagram for you. The decarboxylation. So the breakdown of, yeah, you could see the cytosol region where you could see the glycolysis. The glycolysis took place. Glycolysis taken place. Now what is going to happen? This pyruvate, it's got completed, right? So in the cytosol, the next part, that is decarboxylation of the pyruvic acid. This process usually occur in the mitochondria. This process called as oxidative decarboxylation. This process called as oxidative decarboxylation. So next to the glycolysis, immediately it will not go to the Krebs cycle or the tricarboxylic acid cycle. This will go to the process called as oxidative decarboxylation. So the decarboxylation process takes place. So glycolysis took place in a cytosol or in a cytoplasm, right? Now, coming to this part here, where this oxidative decarboxylation going to take place? This oxidative decarboxylation takes place in the mitochondria. Sometimes they'll ask the site also. The site where the oxidative decarboxylation takes place, it's not in the cytoplasm, because glycolysis in the cytoplasm, but oxidative decarboxylation. What is the oxidative decarboxylation? Where the pyruvic acid, the pyruvate, which is getting converted to what? Which is getting converted to acetyl-CoA. This acetyl-CoA, what will be happening to acetyl-CoA? Acetyl-CoA react with the oxaloacetate. This will react with the oxaloacetate. This will react with the oxaloacetate. From here, the Krebs cycle begins. From here only the Krebs cycle begins. Not immediately with the glycolysis. Once the glycolysis gets over, the pyruvic acid or pyruvate forms, then immediately it go with the oxidative decarboxylation. This process takes place in the mitochondria. But one thing that you should remember, here is NADH2 is getting formed. So here also NADH2 is getting formed. This you should remember. But the time when we calculate the number of ATP, this place you should remember where during oxidative decarboxylation, one NADPH has been formed. It just not one, it means two. Why it is two? Two places where it becomes two pyruvic acid, right? So pyruvic acid at the end of glycolysis. So this pyruvic acid again go with this process, oxidative decarboxylation. Again, this pyruvic acid will go to the oxidative decarboxylation. Here one Krebs cycle takes place. Here one Krebs cycle takes place. So what happens to this acetyl-CoA? This acetyl-CoA which reacts with the oxaloacetate, and then there is a Krebs cycle. The Krebs cycle, the process begins. But question comes like, again ATP after the formation of pyruvic acid, whether they're asking question right from this process, right from here. From the pyruvic acid, you need to include this NADH2. Or they asking questions only from the acetyl-CoA, then you can ignore this NADH. So I already told you like NADH2. So NADH2 is equivalent to 3 ATPs. Then you will be positioned to include like 3, this side 3 and that side 3. There will be like 6 ATPs that you need to include when the question comes from, if they're asking question from the pyruvic acid. If the question comes from how many ATPs from the acetyl-CoA, the question comes because this process occurs before the acetyl-CoA, right? So you can ignore this NADH2. You can ignore this NADH2. Okay. So guys, you please read, because you will be very, very cautious in this particular area where pyruvic acid end of the glycolysis immediately will not go to the Krebs cycle. It go to the oxidative decarboxylation, where what happens? The pyruvic acid with the help of the coenzyme, where the oxygen will be liberated out and this NADH2 will be formed and the acetyl-CoA will be produced. And this acetyl-CoA through the oxaloacetate, this go and merge with the Krebs cycle. And this is the connective link that you should be very careful. Oxidative decarboxylation is a connective link between. This is a connective link between what? It's a connective link between. It's between your glycolysis, between a glycolysis and Krebs cycle. So sometimes the question comes, what's actually the connecting link? The connecting link, they give an option. Electron transport chain, different things, they have oxidative phosphorylation, so options they'll give. But you should go with the oxidative decarboxylation. Oxidative decarboxylation, which acts like a connective link between the glycolysis and the Krebs cycle, because it acts like a connecting bridge between. How? By means of this process. This is one area. And second area, how many ATPs, when you're about to calculate the ATPs production, like from the glycolysis, you must read the question properly, whether the question is from, they're asking after the pyruvic acid or they're asking after the acetyl-CoA. Accordingly, like you can include this NADH2 or you can ignore this NADH2. So when you make it as like in the calculative part, you should be cautious, right? Extra cautious about this particular link, that is oxidative decarboxylation, because here there's no ATP. Here no ATP, no ATP is getting formed, but here you could see NADH2. You ask like, sir, this process occurring in every plant? No, that is like, slight manipulations like modification. But oxidative, if it is like a respiratory plants, if it requires more of oxygen, then mostly it go with the aerobic respiration. Around like a majority of the plants, it goes with this general process. But only the rare exceptions where you could see CAM pathway, like crustacean acid metabolism, there's special modification, there you could see this type of process may not be taken place. But in general, this is the process, the respiration, aerobic respiration takes place in most of the plants. Fine. So you clear with this? First we done with the glycolysis. Second we done with the decarboxylation of the pyruvic acid. So 2-pyruvic acid is produced at the end of the glycolysis. It eventually produced, what is the fate of the decarboxylation? This produced acetyl-CoA. Acetyl-CoA merged with the oxaloacetate and it goes to the Krebs cycle. Now we discussed about the fate. What is the fate of the pyruvic acid? So pyruvic acid being produced out of the glycolysis. So now what is the fate of the pyruvic acid? Here the pyruvic acid, whether in the presence of oxygen or in the absence of oxygen, this can take any route. Whether there is a presence of oxygen, so what happens, like this goes to the, if there is a presence of oxygen, then what happens? Then definitely it go to the aerobic respiration. If there is a presence of oxygen, this goes to the aerobic respiration. If there is an absence of oxygen, if the oxygen is not present, then in the environment, then what happens? This goes to the C2H5OH. C2H5OH. Let me write in a black. So here C2, this is alcoholic for ethyl alcohol, C2H5OH. This happens in the absence of oxygen. If it is in the presence of oxygen, it goes with the aerobic respiration. If it is an aerobic respiration, carbon dioxide and H2O and energy will be produced. Carbon dioxide plus H2O and energy will be produced in case of aerobic respiration. Also, you could see, like along with the alcoholic fermentation, you could see if there is a like absence of oxygen, you could see lactic acid fermentation also takes place. This we already discussed. Lactic acid fermentation takes place. Lactic acid C3H6O3. So C3H6 H6O3. So the lactic acid fermentation. So which part of a body, like for human beings, humans are concerned, where actually the lactic acid are produced, where the lactic acid, which part of like humans where the lactic acid usually get produced? Because if there is accumulation of lactic acid, it clearly tells like there is an absence of oxygen. If you are an athlete, you are running for a long time in a ground, like one point of time what will happen, you started suffocating, you cannot breathe properly. This because of the lactic acid accumulation. Can you tell me which part of the body, human body, where it produces this lactic acid? Yeah, you correct, muscles. Usually in the muscles, there's a reason why, because of the muscle fatigue condition. We discussed in the locomotion and movement, that area, when we're talking about the muscles, we discussed due to the accumulation of the lactic acid. When the lactic acid forms, then there will be a depletion of oxygen because it occurs in the absence of oxygen. So at that point of time, the muscle fatigue, that this type of feature that you could see, then person cannot like run faster, then person need to take a break, and person might have a suffocation due to the lack of oxygen. If the question comes like where the lactic acid produced in the human body, then it is definitely it's a muscle. But if it is produced, then it leads to the anaerobic condition as far as lactic acid is concerned. Fine. So this is the fate of the pyruvic acid. The fate of the pyruvic acid that we seen is a presence of oxygen or in the absence of oxygen. If it is an absence of oxygen, it goes with the alcoholic fermentation, that is ethyl alcohol. In the absence of oxygen, either it go with the lactic acid fermentation. If it is a presence of oxygen, it goes with the aerobic respiration. Even the plants that you asked me whether all the plants which takes up this process, again it depends upon the environment, where the environment, how much of like the oxygen and the carbon dioxide. This depends upon the environment, the oxygen and the carbon dioxide ratio. Based on that, the process takes place. So this is the fate of the pyruvic acid. So far we discussed glycolysis and we discussed the decarboxylation of pyruvic acid and the fate of the pyruvic acid. Fine. So what happens like in the same way, if you could see in case of like an anaerobic respiration, I told you like this is not in the case of aerobic respiration. In anaerobic respiration, you could see only net gain is only 2 ATP. This you should always remember. If it is going with the anaerobic respiration like this by means of lactic acid or alcoholic fermentation or acetic acid fermentation, there you could see only there is ATP, 2 ATPs. 2 ATPs is actually the net gain in the absence of oxygen. In the absence of oxygen or in anaerobic respiration, where you could see this type of thing. So how many ATPs are produced for one sucrose? You need to calculate because we're talking about the glucose. Can you tell me like sucrose is actually what type of saccharide, what type of sugar is this? So usually the ATPs usually produced out of the glucose molecules. When it, because we're studying this process takes place not with a complex form of sugar. This occurs by means of the simple sugar. Simple sugar is actually monosaccharide. It has to be converted to the glucose form. From the glucose, the simpler forms only, the cellular respiration takes place. But the cells, as if it cannot go with the sucrose because it's a complex form of sugar. This respiration process which recommends the monosaccharide, that is glucose. The main recommendation is itself, it must be the monosaccharide. So it must be in a simpler form. Only if it is getting converted to simpler form only, this has to be converted into the pyruvic acid, that is the product. So sucrose, if it comes, it's not a monosaccharide unless and until if it is getting converted into simpler substances. This type of ATP production may not be taking place. Even if it occurs, there also more of energy that is required when compared to the normal glycolysis, which is happening by or from the normal glucose molecules, because glucose is a monosaccharide. So we done with this part, that is decarboxylation. Next we move on to the tricarboxylic acid cycle. So now we moving to the citric acid cycle, that is Krebs cycle. We'll start with the Krebs cycle. Now oxaloacetate has been formed. So this is what actually we are talking about, the pyruvate decarboxylation. This area only talking about decarboxylation. So understand, this is one pyruvate. The same way the other pyruvate is also like is going with the same process. There are two pyruvic acid which is formed. So I just mentioning only one. The same, you need to assume there is one more cycle which is happening on the other side. So both the sides of pyruvic acid which is with the same processes taking place. So what happened now? This is the decarboxylation where the pyruvate is getting converted to acetyl CoA. If it is getting converted to acetyl CoA, that one thing that you should remember is NADH2. This NADH2 is getting formed during this process. That is pyruvate decarboxylation, NADH2, that you should remember. Now what happens? This NADH2, which leads to acetyl-CoA. This acetyl-CoA combines with oxaloacetate, and this enters inside what? Enters inside the Krebs cycle. Now it's going to the Krebs cycle. In this Krebs cycle, so who first found the Krebs cycle? It was actually identified the year 1937. 1937, the Krebs cycle was discovered actually. What's the name of the person who first identified this cycle, tricarboxylic? This is otherwise called as TCA cycle, tricarboxylic acid cycle. TCA stands for tricarboxylic acid cycle, or this is otherwise citric acid cycle, or we also call it as a Krebs cycle. Krebs, yeah, it was identified by Krebs and Hans. Fine. Then where exactly this is taking place, the tricarboxylic acid is taking place, it's happening in a mitochondrial matrix. In eukaryotes, this occurs in a mitochondrial matrix. In case of eukaryotes, but in prokaryotes, this tricarboxylic acid takes place in the cytoplasm itself. That makes a difference. But in case of prokaryotes, TCA cycle takes place in the cytoplasm itself. But in eukaryotes, this takes place on the not in mitochondria, it's a mitochondrial matrix. This particular process takes place. So what is happening with the TCA cycle, where one molecules of the pyruvic acid is going with the 3 decarboxylation and 5 oxidation, and then eventually what will happen? 2 molecules of pyruvic acid which is producing 2 molecules of ATP. Can someone tell me, like, how many ATPs are got produced in the citric acid cycle? How many ATP that you could see in a citric acid cycle? Can you sum up with this complete reaction? How many ATP and how many NADH2 and how many FADH2? So when we discussed actually in case of glycolysis, right? The same way, when we try to consolidate the citric acid cycle, how many ATP, how many NADPH2 and how many FADH2? Because FADH2 was not there in a glycolysis, but in Krebs cycle when we discussed, you could see FADH2. So here you could see the FADH2. In this place you could see FADH2 that you could see, where the succinate which gets converted to the fumarate, you could see FAD, which is by means of succinate dehydrogenase, you could see one FADH2. So we come to that part, the consolidating part. So how, like, Krebs cycle, they asking about one Krebs cycle or they ask about the 2 Krebs cycle? We read the question and then we're going to answer this particular question. And coming to here, this part, here NADH2, here is one NADH2, here you could see NADH2, and here you could see NADH2. So here NADH2, here this is the third place that you could see NADH2. And ATP, where is an ATP here? Can someone tell me, like, where is an ATP? Which part where you could see the Krebs cycle? Is there any ATP here in this particular? But here in this case, this is getting produced in the form of GTP, that is guanosine triphosphate. This is not directly produced in the form of ATP. In Krebs cycle, it produced in the form of what? Produced in the form of the guanosine diphosphate, which get converted to guanosine triphosphate. So this GTP with this produce ATP. So this place you could see one ATP, but here it is in the form of GTP. It is a form of GTP, but GTP is also playing the same functions like that of ATP, but this converts, the GTPs get converted to the ATP. So coming to cycle, citrate, then we have isocitrate, then alpha ketoglutarate, succinyl-CoA, then the succinate, the fumarate, malate, oxaloacetate. So again the same cycle continues. So assume the same cycle which is happening, like, the other side also, the one more cycle, that citric acid cycle, which is going to take in place. So now we go to the consolidation part, like how many ATPs, NADH2, FADH2, that we discussed already in case of glycolysis. Now we come to the Krebs cycle. So we discussed about the Krebs cycle. Coming to the Krebs cycle, we discussed how many ATP and NADH2 and FADH2. So we have 3 NADH2. Okay. Then how many FADH2? We have 1 FADH2 and we have ATP. So we have ATP is actually 1. So this is what actually the Krebs cycle we're talking about. So 3 NADH2, 1 FADH2, 1 ATP. So as I already told you, 1 NADPH2 is equivalent to what? 1 NADPH2 is equal to 3. So 3 into 3. And 1 FADH2 is equal to 2. So 1 into 2. So this gives 9 and this gives 4 and this gives 1. Okay, so 1 NF, so example. So here it is actually 1 FADH2, right? So 1 FADH2 is actually 1 into 2 is actually 2. Here it is 9 and it is 1. So when you calculate like 3 NADH2, 1 FADH2 and 1 ATP, these all things that gives 12 ATP. So altogether this gives 12 ATP. This is for one cycle. I mean one pyruvic acid, how it's getting converted for one. But since it's happening on the both the sides, right? So this 12 ATP has to be multiplied into 2 because 2 pyruvic acid. So 12 into 2, this gives 24 ATP. So the net gain which is happening in this case here is 24. Guys, you please understand Krebs cycle, and we discussed about one Krebs cycle. Understand there are two pyruvic acid, the same thing which is happening in two sides. So you need to multiply the gain, the 12 ATP for one Krebs cycle because it's happening on both the sides for two pyruvic acid. So 12 ATP into 2, this will give the 24 ATP. How the calculation? 1 NADH2 is equal to 3 ATP. 1 FADH2 is equivalent to 2 ATP. So if you multiply, so 9 and 9 plus 2 plus 1 is for 12 per cycle, and 12 into 2 that you just multiply, this gives 24 ATP. But question comes where, like, after acetyl CoA or from the pyruvic acid that I already told you. If the question comes like right from the pyruvic acid, then how many ATPs that you need to include? How many ATPs? For example, this is happening after the acetyl CoA, only with the Krebs cycle. But the question comes right from the pyruvic acid, right from the pyruvic acid. What happened? You need to include the pyruvate decarboxylation. Decarboxylation. That process also you need to include. Guys, please pay attention. Be very cautious. When you question, they are asking about only after the acetyl CoA, only with the Krebs cycle. This is the correct 24 ATP. If the question comes right from the pyruvic acid, then from the pyruvic acid, if they are asking you to calculate the total number of ATP, then you must include the number of ATPs produced during the pyruvate decarboxylation also. Then can you tell me how many ATPs is supposed to include, or you need to add along with this 24 ATP? Can someone tell me? Just only with the Krebs cycle, that is with the after the acetyl CoA, you could see the 24 ATP. This is the correct answer. The 24 is the correct answer. But the question comes, need to include right from what? Right from the pyruvic acid. You need to include the pyruvate decarboxylation itself. That means before the formation of the acetyl CoA, that process also that you need to include. That point, they need to include how many ATPs has to be summed up. How many ATPs that you need to include? So what is actually the total ATP that you can get if you include the pyruvate decarboxylation? Because pyruvate decarboxylation, there are like 6 more ATP. So if you add 6 more ATP, so 24 plus 6, this will give 30 ATP. So this area you need to be double cautious. When you read the question, blindly don't answer. So please read the question properly. Because how the 6 came? Because the 6 came from, because one cycle, that is NADPH2, 1 NADPH2 is equal to 3, 2 cycles. So 3 plus 3. So definitely 3 plus 3 is equal to 6 ATP. You need to include the 6 ATP. So the answer will be 30. So when you need to answer 30, when you need to pick up the option 24, you need to be very careful. So only after the acetyl CoA, that is only you're talking about the citric acid cycle, 24 ATP. But you are including right from after the pyruvic acid formation, immediately after the glycolysis, you are including pyruvate decarboxylation, then you must include 6 ATPs with this 24, then the total gain of ATP is 30. Clear, guys? So we're done with two parts, that is glycolysis and the Krebs cycle. But when the question comes only about the pyruvate decarboxylation, the answer is only the 6 ATP. That also you should be very careful. Only with the pyruvate decarboxylation, if any question comes, it's only the 6 ATP. So far, here in glycolysis, the net gain is 2 ATP. There are remaining the 6 ATP is there, but it's not an encashed. I told you only in the oxidative decarboxylation, this check will be encashed. So now the ATP has not been consumed. Okay, so only this 2 ATP is a net gain, and pyruvate decarboxylation 6 ATP been produced, and Krebs cycle 24 ATP. So now you could see 24 plus 6 together we have a 30 ATPs. So this calculative part you should be very careful if they together, because you told like 36 ATPs together, right? So when you like split up and they ask how many ATPs in each and every aerobic respiration, you must be in a position to answer. So for that, you read the question properly, and then you need to be extra cautious, and then you answer this type of question. I hope you guys understood. So glycolysis, and we discussed about the Krebs cycle. Then we move to the third part is the electron transport chain. Coming to the electron transport chain, so the third type is electron transport chain. In the electron transport chain, so what's going to happen? It's usually here the generation of ATP is going to take place. So the generation of ATP is going to take place in the electron transport chain from the molecule of the hydrogen, that is through the proton acceptor. Through what? Through the proton acceptor. Through the proton acceptor, this is going to take place. Fine. Now here you could see with the help of NADH2, with the help of FADH2, this electron transport system is going to take place. Where this is going to take place? In inner mitochondrial membrane. In an inner mitochondrial, they ask for the location. This electron transport chain occurs in a inner mitochondrial membrane. This consists of series of stages. We have the 5 complex here: complex 1, complex 2, 3, 4 and 5. This consists of series of 5 complexes together. We call it as electron transport chain. So we have the shuttles here. This shuttles which carries what? Which carries the electron. So which acts like an electron carriers. Which acts like electron carriers. And this electron carriers which moves from the one shuttle to the other shuttles. And this is the place where the carriers, like the electron carriers, which will be acting like a shuttles. And here we have 2 shuttle systems. Can someone tell me what are the 2 types of shuttle systems that you could see? So we have the shuttles here, 2 shuttle system. Because I'm not explaining in detail about each and every complex, because every complex, the complex details, everything you would have studied in the textbook itself. So how like one complex to other complexes, electrons like you're going to get, the carriers going to get disseminated, that you studied already. But I just explained now about the 2 shuttle systems usually which is involved in a 2 shuttle systems involved in this type of the electron transport. So the 2 shuttle system is, one is called as malate aspartate. We call it as a malate aspartate. The second type of shuttle system, the second shuttle system is glycerol phosphate. The second is a glycerol phosphate. So these 2 types of shuttle system which is actually involved in the electron transport system, this is responsible for transferring. So what makes the difference between the malate aspartate and the glycerol phosphate? Malate aspartate, here it is no loss of ATP. In this case, there is no loss of ATP. The difference. But here in glycerol phosphate shuttle system, there is a loss of 1 ATP. Loss of 1 ATP. So which shuttle system is very effective? We're going to discuss about the shuttle system. I just explained like how this like ATP is gaining transfer. So either we go with the malate aspartate shuttle system, or it can go with the glycerol phosphate shuttle system, right? Which seems to be efficient? Based on this, you can tell me malate aspartate shuttle system, there is no loss of ATP. But in case of glycerol phosphate shuttle, there is a loss of 1 ATP. Can you now tell me which shuttle system which seems to be effective? Having this like gain and a loss of ATP, can you tell me which shuttle system it seems to be more efficient? Okay, so now we move to the shuttle systems. I told you like malate aspartate system and the glycerol phosphate system. So first we'll discuss about the glycerol shuttle system, and we discuss about malate shuttle system. Coming to the glycerol, this is a cytoplasm and this is a mitochondrial membrane that you take. This is a cytoplasm. This is a cytoplasm of a cell, and this is the mitochondrial membrane. And so what is happening here in this case? Here you could see 2NADH2 is getting converted to NADH, and this 2H is getting transferred, and this requires ATP. So here plus FAD, here FADH2. will be formed in case of glycerol. So FADH2 is getting formed. But you take in the same way in case of malate aspartate system. This is again the cytoplasm. This is a mitochondrial membrane that you could take. In case of malate aspartate system, here the same mechanism to 2 NADH2, like there also NADH2. So here also like 2 NADH2. Here you could see NADH. So it's getting transferred. So what you could see the difference between the glycerol, this shuttle system, and the malate system. There you could see 2H plus NAD. This gives to NADH2. NADH2. So I told you already, one FADH2 is equal to how many ATP? I'm going to just give a break. Don't worry. With this, I'll give a break. One FADH2 is equal to how many ATPs? There are 2 ATP, because one FADH2 is equal to 2 ATP. FADH2. One NADH2 is equal to, here it is going to 3 ATP. So where you could see more of ATPs in case of malate only. So malate only you could see more of ATP. So this seems to be efficient. This seems to be efficient because it's producing NADH2, because one NADH2 is equivalent to 3 ATP, but one FADH2 is equal to 2 ATP. Here only 2 ATP is getting produced out of this shuttle system, glycerol shuttle system. But in case of malate shuttle system, and there is no loss of ATPs also, but you are getting additional 1 ATP. You could see 3 ATPs here. So malate shuttle system, which confirmed to be more effective than what, than this thing. And now you ask on questions like, there are 2 ATPs, like, which is getting consumed in the type of glycolysis. Can you tell me, like, where exactly the 2 ATPs which is going to get consumed? Okay, whether it's going to get consumed in this shuttle system or it's going to get consumed in the oxidative phosphorylation. This clarifications we'll have after the break. So now the time is 3:35. Just give you a 5 minutes break. Come back by 3:40. You can go for a break, a short break. Welcome back, students. So let us begin the session. So we are discussing about the electron transport chain, right? In the electron transport chain, we discussed two types of shuttle system. One is malate aspartate, malate shuttle anti-aspartate, where we confirm the malate seems to be like more efficient because there is no loss of ATP, and malate which seems to be three ATP molecules which is getting produced. So due to this reasons, malate shuttle system which seems to be more effective than the other one. Fine. So now coming to the components of the electron transport chain. So we discussed about the constituents. What are the main constituents that you could see in the electron transport chain? Constituents of electron transport chain. The first and foremost thing is NAD. NAD, we have FMN, FAD, then coenzyme, Q, and we have cytochrome. We have different cytochrome. So these are the different components. You know the expansion for NAD, nicotinamide adenine dinucleotide, FM and flavin mononucleotide, flavin adenine dinucleotide FAD, coenzyme Q. What is the other name for coenzyme Q? What is the other name for coenzyme Q? And what is the cytochrome? Cytochromes, like you could see B1, C1, B1, C1, C. These are the different cytochromes that you could see. These are the different constituents which is present along with cytochrome A, A3. These are the different constituents you could see in case of electron transport chain. Okay, so coenzyme Q can also be called as ubiquinone. This can be also called as ubiquinone. Ubiquinone is other name for coenzyme Q. Fine. So these are the constituents which assemble and coordinate in such a way, like through the electron carriers, like by means of this shuttle system, two shuttle system, with the 5 complex, how this effective mechanism is going to take place. But many of the textbooks, they would have given only this till 4 actually, and we discussed separately the 5th complex. This 5th complex we discussed separately because this 5th complex, we call it as an oxidative phosphorylation. This takes place by means of FOF1. These units. This takes place by means of FOF1 units by means of an enzyme called as ATP synthase. By means of ATP synthase. This 5th complex, where we call it as oxidative phosphorylation, takes place. This process is called as oxidative phosphorylation. Phosphorylation. The last segment of the electron transport chain that we'll see. So how many complexes are there totally? The 5 complexes. How many shuttle system? We have 2 shuttle system. You know which shuttle system is more effective and what exact process which is taking place where the electron, through the electron carriers, the protons is getting pumped right from the matrix to the inner space, and this process which takes place in the inner mitochondrial membrane. Now we'll see this magnified view of this particular, the 5th complex, that is oxidative phosphorylation. And you should understand the oxidative phosphorylation where the 2 ATPs, we discussed like 38 ATPs, right? Out of 38, the 2 ATPs which are consumed, we discussed, we cannot forget this, right? So 38 minus 2, that only which gives the total gain of the aerobic respiration is the 36. So what happened? You asked this question, what happened where these 2 ATPs got lost? So it's actually lost in this process. There's an oxidative phosphorylation. In this oxidative phosphorylation, where these 2 ATP molecules got lost, that we're going to discuss in the separate process where oxidative phosphorylation. So this is a magnified image that you would have studied in the chemiosmotic hypothesis also. Yesterday also we discussed, but we didn't discuss much about this ATP synthase, this particular complex. Now we'll see this oxidative phosphorylation, how it takes place. This is actually like an acceptance of electrons at the end of the respiratory chain where the terminal oxidation takes place. This is a place where the terminal oxidation takes place. So this is inner mitochondrial membrane. It's the same thing like what we studied in the electron transport chain, the same thing that we have discussed, going to discuss here. Here you could see the 2 units here. Here we have a F4 unit and here we have a F1 unit. This unit is called as F1 and we have a F4 F1. So that we call it as this complex as like F4 F1 unit or F4 F1 complex. But What is happening now is actually the 2H, this protons has to be pumped out. Even yesterday we discussed, when we discussed about chemiosmotic hypothesis, we discussed about photolysis of water, how this equilibrium, the constant is getting maintained when there is accumulation of the 2H molecules inside the lumen, that is inner thylakoid membrane. It needs to be pumped out. How it's getting pumped out? It's actually pumped out by means of this complex F1 complex. Here you could see an enzyme. This enzyme is called as ATP synthase. ATP synthase. This ATP synthase, what it will do is actually when it's about to pump this to, because it's impermeable, where it requires energy, right? For that, like it from inside, it should come out of this structure. So it requires this enzyme, ATP synthase enzyme. This ATP synthase, which combines like adenosine diphosphate and the phosphate molecules with the F1, and this produce ATP. So this is how the ATP molecules getting used up. So out of 38, two ATP molecules getting used up where? In this process, oxidative phosphorylation, where the two ATP molecules getting wasted up. So that is the reason, like we mentioned, like 38 minus 2. And this gives the 36 ATP, it's a gain. So where actually this two ATPs which is getting used up, this two ATP is getting used up in this F1, that is an oxidative phosphorylation, where it's because when it's pumping these two proton molecules, it requires an energy. By means of the enzyme, what happens? This produces in the F1 unit, it produces by combining ADP with the phosphate, this producing and utilizing ATP. Here, two ATP molecules getting used up. So it's getting used up. So you can now clearly identify where the two ATP is getting lost out of the 38. Oxidative phosphorylation. This process we call it as an oxidative phosphorylation. Oxidative phosphorylation. By means of this process, what happens, like this usually occurs in where, in which type of shuttle system? If you just clearly understood the shuttle system, where, which type of shuttle system this is actually happening, because we done with the two shuttle system. Which shuttle system where it takes up or utilize the ATP? Now you must be in a position to answer which type of shuttle system which requires an ATP, so that this process occurs in that particular shuttle system, because we discussed which one is effective already. So in that particular system where it requires an energy, then what happens, when it transfers, this protons getting transferred, it requires an energy. So ATP synthase, which is getting produced out of this FO, where this takes the ATP is getting converted to ATP, so ATP molecules getting used up here. So where this is happening, oxidative phosphorylation, this is happening in an inner mitochondrial membrane. This happening in an inner mitochondrial membrane. Question comes, where the oxidative phosphorylation takes place? This takes place in the inner mitochondrial membrane in eukaryotes. But prokaryotes, can you tell me where, in which part of the prokaryotic cell this process takes place? Oxidative phosphorylation process, which takes place in inner mitochondrial membrane as far as eukaryotes. But in case of prokaryote, can you tell me which part of the prokaryotic cell where this process takes place? That is oxidative, but this will not take place in a what is it called, the inner mitochondrial membrane. It will not take place there. There's a specific location where you could see in prokaryotes this takes place. This takes place in a mesosomes. In prokaryotic cell, there are inclusions you could see. It's not exactly the cytoplasm. We have inclusions which present in the cytoplasm. In that inclusions, this process where the oxidative phosphorylation takes place as far as prokaryotes are concerned. So this is the place. Now you solved out of 38 ATP, the two ATPs utilized. The loss actually which occur in which phase? This is an oxidative phosphorylation. So all the remaining 36 ATP, these all seems to be the gain for you. So we're done with oxidative phosphorylation. Now, further confirmations of the oxidative phosphorylation, we'll just continue with yesterday we discussed one part. Do you remember chemiosmotic hypothesis? So can you recollect yesterday in photosynthesis also we discussed? Let us correlate. This is what actually we're talking about, oxidative phosphorylation. Can you now recollect? This is the part we're talking about, oxidative phosphorylation. Oxidative phosphorylation. This is what is happening actually. Yesterday we discussed this hypothesis called as chemiosmotic hypothesis. So I told you already, electron transport chain. Electron transport chain which takes place in the inner mitochondrial matrix. Inner mitochondrial matrix. Okay, so this is a chemiosmotic hypothesis. Who proposed chemi-osmotic hypothesis? Who proposed chemi-osmotic hypothesis? Can you tell me the name of the person who proposed chemi-osmotic hypothesis? This is proposed by Peter. It's proposed by the Peter Mitchell. We call it as Peter Mitchell. Peter Mitchell. The chemi-osmotic hypothesis was proposed by Peter Mitchell in the 1961. So you asked me the question, electron transport chain. Yes, it is. Then mitochondrial membrane, inner membrane of the mitochondria, this takes place, electron transport chain. But oxidative phosphorylation takes place in the inner membrane, the matrix of the inner membrane. So Peter Mitchell, he proposed in the year 1960. Are you sure? 1966. Please confirm it. It's like mentioned as 1961. Kirti, can you please confirm it is 1966 or it is 61? Because I could see it's 1961 here. Anyway, we'll confirm, but I don't know. But as per my reference, it shows 1961. I'll just confirm it. So Peter Mitchell performed 1961 this chemi-osmotic hypothesis. Fine. So this is what actually we discussed yesterday about the what is that called the photosystems, the photosynthesis part we're talking about, the thylakoid membrane. Then we have the inner membrane. This we have the inner membrane, the lumen, everything we discussed. And this is the part that photolysis takes place. So the loss of water, the photolysis take place. Do you remember I told you yesterday, even the photolysis, three important things where you could see when the photolysis, we could see half molecule of nascent oxygen, we have hydrogen, and we have electrons. And these electrons, which is going to get pumped from one photosystem to the other photosystem through the plastoquinone, right? So what will be the fate of this, the hydrogen? This will be getting clumped inside actually. So when it is getting clumped, and also the plastoquinone is also going to bring actually the hydrogen ions inside. When there is a density of this hydrogen, which seems to be more, yes, are you clear? Do you confirm it's 1961? Then we confirm, we freeze 1961 itself, because my reference shows it is 1961 only. So can we freeze with 1961? Fine. So this is a proton, the protons that you could see. So when there is density of the concentration of these protons which is getting accumulated inside, then it need to be pumped out of this the central part of the inner part of the mitochondrial membrane to outside to the stroma. For that it requires energy. This is what we're talking about. This is the F4 part and this is the F1 part, and this is the part where oxidative phosphorylation takes place. And by means of ATP synthase enzyme, the ADP is getting converted to ATP. So now the ATP has been formed. Fine, the ATP has been formed. So here, this is the place where the ATP is getting consumed in oxidative phosphorylation. So these two ATP, the two ATPs getting consumed in this part, oxidative phosphorylation. So this is the confirmations where the out of 38, 2 ATPs where it is getting lost or used up, it's actually used up in the oxidative phosphorylation. Okay, so this is a confirmative link with the photolysis of water, where there is a loss of water, and with the usage of energy ATP, and with the oxidative phosphorylation. So it's a connecting spot that image shows between the photosynthesis part and the respiration, that is chemi-osmotic. And you guys asked me, the previous someone asked me, is there difference between the plants, the cycles, the C2 cycle, C3 cycle? Yes, of course, because it depends upon the how much of oxygen it takes up, how much the intensity of the photolysis takes place, whether the plants takes more amount of water, the plants are temperate plants, or plants are succulent plants or xerophytes. So this amount of, if at all if the plant cannot spend much of energy, then it can go with a modified type of pathways that we discussed, like C4 pathway, or it can go with a crustacean acid metabolic pathway. Or else what happens most of the times, carbon dioxide will be get wasted in terms of photorespiration. Whether the plants can go with the special sort of adaptive measures, or the plants need to spend more of energy for taking this much of, or fulfill the needs actually. So it keep varying from the plants to plants, but in general, normal cases where you could see the 38 ATPs and out of the 1 glucose molecules, where out of 38 ATPs, the 2 ATPs which is getting consumed in case of oxidative phosphorylation, the remaining 36 ATP, it seems to be a gain for aerobic respiration. Okay, this again depends upon the type of the adaptation where the plant has got, and where the plant has been rooted, and where it's actually is growing. This also the adaptive criteria also that we need to take in for consideration. So We done with chemi-osmotic thing. Then we move on to the next part. The next part is a exose monophosphate pathway. So can you someone tell me what's the other name for exose monophosphate pathway? Exose monophosphate. This is a exose monophosphate pathway. Exose monophosphate pathway. So simply we call it as HMP shunt. This is also called as shunt, exose monophosphate pathway, or this also called as triple P. PPP we call it as, or it can be also called as triple P. The triple P is a pentose phosphate pathway. So oxidative phosphorylation. So since you could see the oxidative phosphorylation where there is a loss of ATP, where you could see the loss of ATP. I told two shuttle systems, right? One is malate, other one is the other shuttle system. You see which one is not effective. That is not effective means because it's using up ATP, it's spending ATP. So that which comes under the oxidative phosphorylation. Which is effective? The effective shuttle system will not come to the oxidative phosphorylation because there is no loss of water here. Fine. So PPP is a pentose phosphate pathway, and this pathway first identified by Warnberg and Dickens. Warnberg and Dickens. In the year, I guess it is in 1938. I just confirm it. Okay, mostly the Kirti, I think. So Kirti, are you there? Kirti, can you tell me? 1938 is correct. Fine. So Warnberg and Dickens. Warnberg, yeah, glycerate. Yes, actually, usually the oxidative phosphorylation and the glycerate thing, but because malate, which seems to be more effective because there's no loss of ATP in malate, but glycerate, this type of shuttle system, where you could see oxidative phosphorylation. Fine. Fine. It's first found by Warnberg and the Dickens in the year 1938. Fine. So this occurs where this process occurs. This is actually shunt of glycolysis. It's an alternative pathway. If the glycolysis is not the compulsory, it need to go with the glycolysis. There's a shunt mechanism. Which usually occurs. This is actually the glycolysis. Actually, it's a bypass mechanisms where you could see this pathway. So we call it as an alternative for the glycolysis. So shunt of glycolysis, or we call it as an HMP shunt. This process usually occurs in a cytoplasm. This occurs in a cytoplasm of a cell. This occurs cytoplasm of a cell, and this is an alternative method of aerobic respiration. This is an alternative method of, this is an alternative method of what? Of aerobic respiration. Aerobic respiration. So that only we call it as an glycolysis shunt, or we call it as an HMP shunt. For this particular thing where it requires more amount of NADP, So this requirement for this triple P, phosphate pentose pathway, right? For that, it requires like more amount of NADP. This usually occurs in a two phases. What are the two phases which is involved? Oxidative phase and the non-oxidative phase. This involves in a oxidative and non-oxidative. There are two phases which involve in this HMP. Oxidative phase, the other one is a non-oxidative phase. Yeah, you can say it's a Warnberg Dickens Lippmann pathway. That is also correct. WDL. Yeah, that is also correct. Lippmann also. Fine. You can add Lippmann. Sorry, you were concerned about the scientist Lippmann. So let me not ignore this person. Lippmann also. Warnberg, Dickens, and also Lippmann. Because he also contributed for this. So this consists of two pathways: oxidative pathway and a non-oxidative pathway. But in this case, what is the total gain of ATP? Can someone tell me what is the total gain? The gain of ATP. In case of normal the aerobic respiration, you could see the 36 is the total gain, the 2 loss of ATP. So we have 38 minus 2, that is 36. So what is the total gain of ATP in the exose monophosphate pathway? So what's the total number of, the amount of ATP? It actually seems to be the net gain of ATP. Net gain of ATP in case of this pathway. Can someone tell me what's the net gain of ATP, ATP in exose monophosphate pathway. Fine. So 24 ATP. So the net gain that you could see in this case is 35 ATP. Here it is a 35 ATP. 35 ATPs that you could see in a exose monophosphate pathway. It's a net gain. Okay. I'm talking about the complete this part of this part of alternative path of glycolysis which involves this alternative pathways where you could see the production of say pseudoeptulose, purine and pyrimidine metabolism, and also which goes to the glyceraldehyde. So combined together, this comes 35 ATPs. So now we discuss about what are the factors which affects aerobic respiration. Factors which affects the respiration. Can you tell me what are the main factors which affects aerobic respiration? Factors affect aerobic respiration. The prime factors, 35 is the correct answer. I told you 35, Kirti, Narmada, you're all correct. You gave the correct answer. 35 ATP is the correct answer for exose monophosphate pathway, the net gain. Coming to the factors is oxygen concentration. The oxygen concentration, it must be below the 5%. As far as the factor, the aerobic concentration, I mean the oxygen concentration, it must be below 5%. And also the respiratory rate, that is carbon dioxide. So that is also a determinant factor. And optimum light, the light source also, which plays a very important role for aerobic respiration. Then you could see ions like magnesium ions, then you could see copper ions. These copper ion, magnesium ions, which also influence the aerobic respiration. And if there is a dehydration, there is a loss of water, then the aerobic respiration, the rate will be decreased. Aerobic respiration rate will be decreased if there is a dehydration. Fine. Next, age of the tissue. If it has become very old, the tissue has become old, and the amount of the substrate. So these are the determinant factors. If the age of the tissue seems to be old age tissues, then again that aerobic respiration which drops down. So deficiency of oxygen. So these are the factors which determine the aerobic respiration. So oxygen concentration, it must be below 5%. Respiratory rate, a suitable optimum light source should be there. Ions like magnesium and copper ions. Then dehydration, where the aerobic respiration rate where it reduces in case of dehydration, and the age of a tissue, the tissue's age which again is getting old age, then again, right, it's again, can you tell me what is actually the percentage of the carbon dioxide? I mentioned is actually the percentage seems to be 5 in case of oxygen, but when you come to the carbon dioxide, what is actually determinantal the percentage of the carbon dioxide, the amount of the carbon dioxide that is required, so that this particular amount or the percentage of the carbon dioxide where you could see the aerobic respiration which seems to be beneficial. Can someone tell me what is the percentage of the carbon dioxide? But here you could see the concentration is a 5%. How about in case of carbon dioxide? Fine. So we're done with the factors. Now we move to the next part. For example, you take in case of quantity, if you want to just calculate the quantity in case of aerobic respiration. So one glucose molecule which gives how much of energy? Can someone tell me? One glucose molecule which gives how much of energy, where one glucose molecule can yield. For example, you take this, gives like 686 kilocalories. 686 kilocalories. This is what actually the energy that we talking about as per the quantity one glucose molecule is concerned. Using this, if we calculate the efficiency of aerobic respiration, if we calculate the efficiency of aerobic respiration, if you calculate the efficiency of the aerobic respiration, so ATP molecules, one ATP molecule will yield 34 kilojoules per mole of energy for one ATP molecule. For one ATP molecule, the yield will become this is 34 kilojoules per moles of energy. But coming to the yield of energy, when you're talking about aerobic respiration, in case of aerobic respiration, it is 1292 kilojoules. This 1292 kilojoules is for what? This is for how many ATPs? This is for 38 ATPs. On the whole, for the aerobic respiration, for 38 ATPs, the aerobic respiration, it requires like 1292 for kilojoules. Which just for one ATP, you take like 34 kilojoules moles per energy. For 38 ATPs, it takes like it's 1292 kilojoules per energy. So glucose stores energy. So how much of the glucose which stores energy? Glucose, how much of energy the glucose can store? This can store of 2870 kilojoules. Basing having this values, if you want to calculate the efficiency of the aerobic respiration, then we need to sum up like that is like 1292. So 1292 by 2870 then into 100. So what is the percentage that you usually will get? What is the percentage? So it's actually like how we calculate the efficiency of the aerobic respiration, just a calculative based part. So in this part I explained one ATP molecule, the yield is like 34 kilojoules. That is for one ATP. Total for aerobic respirations together for 38 ATP, where you could see 1292 kilojoules, that is for 38 ATP. Where the glucose stores the energy is actually 2870 kilojoules. You just want to calculate the efficiency. Then what you'll do is 1292, that is like the total aerobic respiration, which divided by the glucose, how much of energy the glucose stores. Then you just calculate and make it into 100. Can you tell me like what is the percentage approximately that you'll get if you just make it's like 1292 by 2870, just you make it and you get like around 45, I think 45 to 47, I think so. Can you tell me it's a 45 or 47? Somewhere it is 45 to 47. You good in maths, guys, can tell me what's the answer that you get? 45 or 46 or 47? How much you get? So 1292 by 2870 that you multiply into 100, you get around 45 or 46. That will be the efficiency. So if a question comes like what is it, how to calculate the efficiency of the aerobic respiration, these values which seems to be beneficial for you. So beneficial is yes 45, right? Correct. So this comes 45%. So efficiency of the aerobic respiration is 45. So how we got this 45? We got by the total ATP molecule yield is for one ATP is 34, right? Total aerobic respirations together with 38 ATP is 1292. How much of like the energy where the glucose can store is 2870. When you just 1292 by 2870, when you calculate, this comes like 45%. This is for your aerobic respiration. So if this seems to be effective, aerobic respiration is effective, then your percentages like efficiency will shows a 45%. If anything which comes down lesser than this 45%, then you can directly confirm the aerobic respiration is not up to the mark. There is somehow that by means of energy, there's somehow in the forms of like ATP malfunction being occurred, then definitely the efficiency of the aerobic respiration also went down. So this percentage is a determinant factor by which we can identify the exactly like the accurate and proper aerobic respiration, whether it's taking place or not. So this is the calculation part by which the efficiency of aerobic respiration takes place. Okay, so question comes like 45% of the what is the efficiency of the aerobic respiration? You just look into the option, it is like 45, then you just go with the correct option. Clear, guys? So we're done with the efficiency of the aerobic respiration. Now we move to the last part, that is the balance sheet. When talking about the balance sheet, before going to the balance sheet, we close with amphibolic pathway. We would have studied in the textbook itself. I'm not going to teach the detail amphibolic pathway. 36, the net gain of ATP in aerobic respiration gain is 36 because 2 lost, right? 2 lost in oxidative phosphorylation. So 2 lost already. So gain is gross is 38. Gross is 38. But gain, when you talk about the use of gross salary, net salary, right? So how much of salary is credited? Because after all the deductions and the PF and ESA, everything is gone off. Like what's it going to take home, right? That is a net. So here the net is 36, but gross is 38. Okay. So coming to amphibolic pathway, here the amphibolic pathways we categorize to fats, carbohydrates, and we have a protein. This amphibolic pathway, the fats that you could see, production of fatty acids and the glycerol here, and the carbohydrates, the simple sugar. And the proteins, you could see the amino acids, and the carbohydrates, you could see glucose 6-phosphate, the normal glycolysis process. Eventually, it comes with the glycolysis process where we end up with the pyruvic acid. But the modes are different. Either go with the sugar like carbohydrates, or go with the fats like fatty acids, or go with the amino acids. It may be any form, but eventually, like where it has to get come and mixed up, it need to come and mixed up with the Krebs cycle. That is by means of acetyl CoA. This is a coordinated, like wherever, whether it is a form of a protein, a carbohydrate, or a fat, whichever the process it takes, but eventually this come and mixed up and forms in the which forms acetyl CoA and get inside the Krebs cycle. This pathway, irrespective, it's a protein, carbohydrate, or fat, this lands somewhere with the Krebs cycle. This is called amphibolic pathway. Clear? So the last part, we move on to the balance sheet. Coming to the balance sheet, so we already done with individual parts. So I hope you can recollect the glycolysis and Krebs cycle. So this part, I hope you're clear. Now we're going to consolidate everything. So we just make the balance sheet of how much of ATP is being utilized. So I'll use white. I can use it black. Balance sheet. This is a balance sheet for ATP production. So first we make it as a step. The steps here, this is for ATP production. You take the ATP or just you take GTP, because one place we discussed in a Krebs cycle thing, we discussed about GTP also. So here FADH2. How many FADH2 or NADH2 is produced, and here you can see the total. Fine. So now you take three important places that we're going to discuss. First one is glycolysis. Number two, we discuss about decarboxylation. Decarboxylation of what? Decarboxylation of pyruvic acid. Third one is a Krebs cycle. Krebs cycle. So now you're going to tell me, like, glycolysis, how many ATPs have been formed? 2 ATP. So 2 ATP. How many NADPH2? No FADH2, so only 2 NADH2 here. So 2 NADH2, it means 1 NADH2 is equal to 3, right? So 2 into 3, this is equal to 6. Okay, so here it is 6. Coming to decarboxylation of pyruvic acid, is there any ATP formed in decarboxylation process? No. So there is no ATP formed in the decarb. You know what is decarboxylation? You've studied, like, how the pyruvic acid getting converted to acetyl CoA, which merged with the oxaloacetate of the citric acid cycle. So no ATP here. Coming to this part, FADH2, NADH2, here 2 NADH2. 2 NADH2. Again 2 into 3 is equal to 6 over here. Coming to the Krebs cycle, here you could see 2 ATP. 2 ATP is formed in a Krebs cycle, but here you could see 3 NADH2, then 1 FADH2. So 3 NADH2 and 1 FADH2. So here 3 NADH2, 6 into 3. 6 into 3 is equal to 18. And 1 FADH2. 1 FADH2 is equal to 2 into 2. So 2 into 2 is equal to 4. Remember, we're talking about 2 Krebs cycle. That you should be very careful. I told you already. We're not talking about, that's the reason I kind of multiplied. So here itself I multiplied. So 3 into 2 is a 6. 1 plus 1, that is actually 2, because 2 Krebs cycle. That you should keep in your mind in the calculative part is concerned. So we're done with all the areas that you could see. So here it is 18, and here you could see the 4. So here you could see 6 plus 2, it is 8. Here 6, only the 6 that will come here. And here you could see 18 plus 4, 18, 22. 22 plus 2, this will come 24. So this is 24. So when you calculate 24 plus 6 plus 8, this comes 38 ATP. This is the total, the gross ATP which produced during aerobic respiration. In aerobic respiration, this is the total ATP. But what happened, that was actually out of this 38, 2 ATP by used up by the pump mechanism, that NADH2, that oxidative phosphorylation mechanisms, where the 2 ATP is being utilized, right? So 38 minus 2, now this will give only 36. So This 2 ATP, where this is got lost, this 2 ATP lost during oxidative. I hope it's not clear, but oxidative phosphorylation. I told you, like in a chemiosmotic even hypothesis, I told you. During this oxidative phosphorylation, this 2 ATPs got lost by means of the shuttle system, right? So here it is like 3 plus 3. So because 1 NADH2 is equal to 3. 1 NADH2 is equal to, here 1 NADH2 is equal to 3, right? So 3 NADH2. So 3 for one cycle. So 3 plus 3, 6. So 3 for one cycle. So 3 for one cycle. So 2 cycles, right? So 3 plus 3 is equal to 6. So 6 into 3 is equal to 18. Okay, so this is how actually, so guys, you got this question how I got 18, because one cycle it is 3 NADH2, one cycle it is 3 NADH2. So you have the 2 cycles, right? So 2 cycles and 1 NADH2 is equal to 3 ATPs. We consider the 3 ATPs. So we need to multiply. 1 NADH2 is equivalent to 3 ATP. We have 2 cycles. So 3 plus 3 is equal to 6. So 6 into 3 that we multiply, we get a 18. So this is how actually, like the balance sheet, the individual part. Sometimes you get a question, the total ATPs for the glycolysis 8, but decarboxylation pyruvic acid, so no ATP, here it is only 6. And Krebs cycle, here you could see 2 ATP and 3 NADH2 and FADH2. Remember, 1 NADH2 is equal to 3 ATP, 1 FADH2 is equivalent to 2 ATP. So I hope you're done with the, I mean, you can understand the calculation. So 36 ATP, what will do? So now this 36 ATP is responsible for all the metabolic and all the reactions of the metabolic reactions and pathways, everything, right? So the energy, because the energy has been considered one, this is for one glucose molecule. Understand, this is for one glucose molecule. So how much ever of glucose that has been taken, you're moving the plants, like, you know, there are a lot of mechanisms like transpiration, pull, photosynthesis, like many different mechanisms, metabolic reactions, enzyme productions, pathways, everything is going to happen. Just for one glucose molecule, this 36 ATP molecules are produced. This is going to get energy for all the metabolic reactions which is going to take place inside the body for one glucose molecule. The same way, how much ever the glucose molecules is going inside, so each and every glucose molecules which go to this aerobic respiration and produce 36 ATP, right? So eventually the plants, it's all like, you know, whether the plant is going to get benefited or not. So it's going to get benefit because we're just giving 2 ATPs, but it's gaining, like, you know, by just only giving 2. But the gain is what? Gain is 36 ATP the plant is getting. But sometimes what will happen, the amount of the energy, if it is more energy need to spend more, means there will be like loss of ATP. Then eventually, like, you know, the remaining thing that is actually received by the plants, which comparatively it is less. So we're done with the balance sheet. The last part we discussed about the respiratory quotient. Coming to the respiratory quotient. So respiratory RQ stands for respiratory quotient. Respiratory quotient. So this made by the formula that we have for the respiratory quotient. Here the RQ, which can be denoted by volume of carbon dioxide, by volume of oxygen absorbed, and this is the carbon dioxide evolved. So this tells you respiratory quotient is actually the volume of the carbon dioxide evolved by the volume of carbon dioxide absorbed. This keeps varying actually from, for example, you take over for the balanced diet, or you take even, like, you know, various proportions like carbohydrates, protein, fats. This keeps varying whether this RQ, which is actually 1 or it is less than 1, that makes the difference actually. For example, you take in case of carbohydrates. You take up carbohydrates where the volume of carbon dioxide evolved is 6 CO2. You take in the form of potato, you eating potato, you eat a rice or whatever it might be, carbohydrate rich food, where this 6 CO2, which is by the volume of 6 molecules of oxygen. If you divide like 6 by 6, then it gives the RQ is for carbohydrate is 1. If you take a fat, so if you take a fat, in case of fat, you take where the carbon dioxide is 102. Whereas if you take of oxygen is 145. You take 145 oxygen, definitely this will be coming lesser than 1. but fat is constant. But carbohydrates, the respiratory quotient is 1 because 6 plus 6 by 6, it's 1. In case of fat, you could see the carbon dioxide is 102, but your oxygen is 145. So definitely, like, it's lesser than 1. It's 0.6 or 0.7, but you need to understand it's lesser than 1. So it is 0.7. The same way you take an anaerobic condition. What will be the anaerobic respiration? You take anaerobic respiration. If you take anaerobic respiration where the carbon dioxide, for example, you take two molecules of carbon dioxide where there is nil. The oxygen is nil. Then what is the answer for you in case of anaerobic respiration? In anaerobic respiration. In case of fat, in palmitic acid, that you could get 0.7. In case of protein, what is the RQ for protein? Protein is 0.9. 0.9. And you take in case of like fat you take fats, it's 0.7. Sometimes you'll get 0.6. The fats will be in this range actually. If you take like organic acids, organic acid, you take organic acids usually it will be the greater than 1. And anaerobic respiration, this usually infinite. Usually it is infinity. Infinity is for the anaerobic respiration. Sometimes RQ values they give, and they'll ask you to identify whether this belong to the carbohydrates or fats. But mostly in the fats, you could see it comes less than 1. The fats you take protein, which comes less than 1. Carbohydrate, the respiratory quotient is 1. Organic acid you take, it goes more than 1. In case of anaerobic respiration, the respiratory quotient, which seems to be infinite. So this is the confirmatory part, a concluding part we done with the this part chapter, that is respiration in plants. So almost we're done with this particular chapter, aerobic respiration and anaerobic respiration. So aerobic respiration, we discussed about glycolysis, and we discussed electron transport chain, tricarboxylic acid cycle, then decarboxylation of pyruvic acid, and we discussed about oxidative phosphorylation. Sometimes the question comes like, which enzyme does not catalyze a reverse reaction in glycolysis? If this question, can someone yeah, infinity is possible. Yes, because here it's like it's nullified. It's zero actually in aerobic respirations, but zero oxygen molecules. So definitely it goes with the infinity. The question, for type of questions, glycolysis. Which enzyme does not catalyze a reverse reaction in glycolysis? Can you answer for this question? Can you answer for this question? Which enzyme which does not involve in the reverse reaction in the glycolysis? Can you, because this type of questions that usually we get in a glycolysis. So glycolysis, this area if you take, which enzyme does not catalyze a reverse reaction in a glycolysis? Can someone tell me, like, what's the name of the enzyme? The enzyme which does not catalyze a reverse reaction in the glycolysis. And sometimes the question comes like, what is the net gain of ATP? What is the net gain of ATP in the the pentose phosphate pathway? What is the net gain of ATP? I told you already, the net gain of pentose phosphate pathway. What is the net gain of the ATP? And sometimes the ratio they ask, actually, the ratio between the aerobic and the anaerobic. So the ratio of ATP synthesis in aerobic and anaerobic respiration, that you should remember, guys, 18:1. 18:1. This value also you should be remember. The ratio of a ATP synthesis between aerobic and anaerobic respiration, often they ask this question. The ratio is 18.1. Okay. So sometimes they ask respiratory quotient of glucose. So if they ask the question, I ask the first question, right? Which enzyme does not catalyze a reverse reaction in the glycolysis? The answer is the pyruvate kinase, which is a common link between aerobic and anaerobic reaction, which is the common link between aerobic and anaerobic reaction. You got the question? Which is the common link between aerobic and anaerobic? If the question comes, which is the connecting link between your glycolysis and the Krebs cycle, the answer is different, right? Which is the connecting link between the aerobic and anaerobic respiration? If this type of connecting link questions comes also, you must be in a position to answer. So you can answer kinase. You just look into the option for first question I asked you, right? Which does not catalyze a reverse reaction in glycolysis, usually go for a kinase enzyme. And the net gain of ATP in glycolysis is actually 8 in number. Net gain of ATP, the net gain of ATP in glycolysis is 8. But if they're more specific in asking a question, how much it's getting used up, then you need to exclude that 6. If the guys listen, the glycolysis, the net gain of ATP in glycolysis means it's 8 in number, but more precisely they're asking a question, how much out of this gain, how many ATP has been utilized? Then it is actually the 2 have been utilized and the 6 ATP which need to be encashed later on. So that you should be very careful. As of now it is 8, but how many is getting used up means it's only the 2 ATP is being used up. So glycolysis, exactly, glycolysis is a connection between the aerobic and the anaerobic respiration. And who discovered exhaust monophosphate pathway, HMP? I told you already. Who discovered exhaust monophosphate, HMP? Who discovered HMP? And which is called as universal hydrogen acceptor. Sometimes they'll ask a question, which is the universal hydrogen acceptor? They give an option like FAD, NAD, NADPH2. They give a carbon dioxide. They give a different option, right? So which we call it as an universal hydrogen acceptor. If the question comes like which is a universal hydrogen acceptor, you must be in a position to answer. If they ask about the ratio between the aerobic and anaerobic respiration of ATP synthesis, the ratio is this 18 is for aerobic and 1 is for anaerobic respiration. They give 1:18, the different option they'll give, but you need to select 18. They ask about anaerobic and aerobic, means always remember 18 is for aerobic and 1 is for anaerobic. So in this place, read the question properly and answer this type of question. So connecting link between the glycolysis and Krebs cycle, which is a connecting link. If there's a connecting link between the aerobic and anaerobic respiration, glycolysis is the correct answer. If they're asking about the connecting link between the glycolysis and the Krebs cycle, so which is a connecting link is between glycolysis and the Krebs cycle? This type of question also they will ask actually. And the other questions like you can get a respiratory quotient in case of like the fats, and in case of like carbohydrates, this type of question you can ask. That is pyruvic acetyl-CoA, or you ask for for the options like pyruvate decarboxylation of pyruvic acid, or the option is given is like acetyl-CoA, you go with acetyl-CoA, or if the decarboxylation of pyruvic acid, the option is given, that is also fine. Sometimes they'll ask a question for the shuttle system, which among the following is the shuttle system is more effective, whether it's a malate or the aspartate shuttle system. So these are the possible questions. So in tricarboxylic acid cycle, where FAD is involved, this type of question. In TCA cycle, where FAD, flavin amide dinucleotide, in TCA cycle, where FAD is involved, one particular part of the cycle they take and they'll ask a question, then you should identify the particular area of the enzyme. So in tricarboxylic acid cycle, they ask the question where the FAD is involved. Where the FAD is involved, usually it's between the succinate and the fumarate. You're getting? So this type of question. So they take only one particular segment and they'll ask this type of question. So in between the succinate and the fumarate, you could see the FAD is the flavin adenine dinucleotide is involved. So it means you go with the TCA cycle or glycolysis, whatever the thing that you're studying, but understand, if you very clear in the particular segments, that will be beneficial. So that type of questions only you get from this chapter. So if you even very clear with the sum up everything, but you don't know like name of the enzymes and these things, like if you couldn't answer, then you may give a incorrect answer. So you might please go through the pathways, especially if I told you like for example universal hydrogen acceptor, it is NAD, that is nicotinamide adenine dinucleotide, is a universal hydrogen acceptor. The answer is NAD. Okay, so who identified from fermentation, like they ask a question like who identified zymase enzyme? We discussed in the previous chapter itself, zymase, we got the Nobel Prize for this, right? In that particular area also, like who's the person who first isolated zymase from the yeast, that is Saccharomyces cerevisiae? What is the name of the scientist? So this type of questions also they give a question. So guys, so these are often this type of question that you'll get. So by this we're done with this chapter, respiration and plants, and tomorrow we'll start with the next chapter, possibly the transport mechanisms and also the nitrogen cycle and nitrogen fixation. So my suggestion for you guys, like practice more amount of number of questions. The more you practice, it makes you perfect. So it's a good time for you. Go for sufficient practice a lot. So there's no limitations for your practice. Please practice a lot, so that will be like beneficial for you, and you get nice good marks and you become a doctor. So I want to see you all like doctors very soon. So I hope you listen to the class and you be safe and sound at home. Thank you so much. I really appreciate your time and patience. Thank you.